Hello everyone. A very good evening to all of you. I hope so. My screen is visible to everyone and the voice is also audible. So I welcome you all to the third day of Brahmastra Series, and I hope so that you all are doing great. You are at the peak of revision because there are very less days left for the examination. Good evening, Malini. So uh, in that case, today we are going to take up Unit 4, which is one of the important unit from examination point of view. And yes, let's take up questions from B, C, and D subunit. So are you all ready with that? Are you all ready to take up the questions? Yes? Okay, let's move on to the question now. This is the most recent question. The signal transduction pathway involved in glycogen metabolism triggered in the liver by the hormone epinephrine that involves the following steps: activation of G protein by the activated receptor, protein kinase A activation, second messenger destruction, adenylyl cyclase activation. Now you need to identify the correct order, the way the signaling is proceeding. So if you can see, these are the options for you, right? A, D, C, B, A, C, D, B. But what I can suggest you, have a look into this question and let me know what is the correct order for this question. Correct signaling sequence for the epinephrine part. So here you have to remember, so epinephrine is related to obviously G protein coupled receptors. So when I say G protein coupled receptors, again you are having different types of GPCRs, right? Identify which GPCR is this. Okay, Malini has already given the answer. Anyone else would like to try on this question? Try on, everyone, because this is not an exam where you are going to lose anything. Whatever you are going to say, end of the day you are going to gain something. If you are wrong, definitely it will be corrected so that you will not make the mistake again. And if you are correct, see, you get that confidence that yes, I have studied something and I remember this pathway well. Yes, participate everyone. Okay, Habib. Anyone else would like to take up the answer for this question? So I can see Malini and Habib has taken the same sequence. Anyone else? Absolutely correct, Malini. So this belongs to the GS pathway. If you remember, we have four major GPCR pathway. What are they? We have GS, we have GQ, GT, and GI. So how you're going to identify this pathway belongs to which category? You have to take into consideration what is the effector protein mentioned over here, what is the protein kinase, what is the second messenger mentioned over here. So based on that, you can easily identify. And always remember, when I say you the sequence, so what is the basic background of any sequence? ADCB is correct. Malini and Habib, both of you are absolutely correct. So when I see the sequence, the ligand goes and binds to the receptor, which in turn activates your effector protein, which in turn leads to the activation of second messenger. Second messenger in turn activates your protein kinase, and this protein kinase is going to phosphorylate the target protein in order to generate the cellular response. And after that, what happens? It will be terminated in any way. Any way in the sense maybe internalization, desensitization, phosphorylation. So we have different methods of termination, right? So this is what going to happen. So overall speaking, if you see, this is the working principle of GPCR protein. So we have an inactive receptor here. So we have the G protein present with 3 subunits bound with GDP and the inactive effector here. So this is, we call it as a resting state. Okay. Now, when a hormone goes and binds to the corresponding receptor, you can see a conformational change occurs over here. See the difference in the image. So, because of this conformational change, it provides the binding site for G protein. So, this G protein goes and binds to the receptor over there. At that time, GDP will be released and GTP will go inside. So when GTP is bound with G protein, what happens? Your alpha, beta, gamma subunit gets separated. So all you have is G alpha GTP. Now this will go and bind to the effector protein because it is having the binding site in the effector protein. So what is the effector protein over here? Adenylyl cyclase. So this will go and bind. Upon binding, your effector becomes activated, ready to carry out the signal transduction. In case if you want to terminate the pathway, what you need to do? You can hydrolyze the GTP. So if GTP is hydrolyzed, obviously it will be terminated. Then there are other ways of termination. Well, in this image, they have provided you with the GTP hydrolysis. Okay. Now, two things to be remembered over here. When your G protein is present in GTP bound state, we call it as an active form. And GTP, you will be having GDP with a gamma phosphate. Remember, the gamma phosphate is stabilized by glycine and threonine in the two switches present on the G protein. And here you can see a scissor sign. So if that gamma phosphate is deleted, in that case, what is retained? GDP. And the protein is in open conformation. So GDP bound will be considered as the inactive state. So remember this basic difference between the bound form and not bound form. Again, what are the enzymes responsible for this catalysis? Remember, guanine exchange factor is responsible for releasing the GDP and taking up the GTP. So you will get the active form. Next, we are having GAP, that is GTPase activating protein, that will undergo hydrolysis of GTP. Now this is present in the GDP bound form. So this becomes the inactive version of the G protein. So remember this part as well as this part. Very important from examination point of view. Every time question do come from here. Okay. So moving on to our next question. CGMP is produced from GTP by the enzyme guanylate cyclase, which exists in the soluble and membrane bound forms. So here are some statements provided. Atrial natriuretic factor, that is ANF, causes natriuresis and diuresis by interacting with membrane bound form of GC. Nitroglycerine causes smooth muscle relaxation and vasodilation by interacting with soluble form of GC. Now, A and D are opposite to each other. Here they have mentioned membrane bound. Here they have mentioned soluble form. So, obviously, one of them has to be correct. Next, the third statement is nitroprusside causes smooth muscle relaxation and vasodilation by interacting with membrane bound of guanylate cyclase. Kinase. So now you need to identify the correct combination of statement here. So basically, when I say cell signaling, you can always expect good number of questions here. And easy to answer, not so tough. Asking you the direct question. 4 marks questions are very frequently asked specifically from GPCR. Try to find out if you are able to get the answer or not. What could be the answer for this? Able to answer it? Are you trying it up? Okay, Malini, I got your answer. A or C got confused. Anupam, okay, 1. Anyone else would like to give the answer for this question? Okay, let me give you the answer along with explanation. So the answer will be, it's not, yeah, it has to be 1 only. It's not 4, it has to be 1. ANF causes natriuresis and diuresis by interacting with membrane-bound form of GC. Nitroglycerin causes relaxation and dilation by binding with the soluble form of guanylate kinase. So obviously if A is correct, D will be incorrect. And how C will be correct? If it was mentioned soluble form. So then it would have been the correct answer. So basically ANF is otherwise known as ANP, is a polypeptide hormone being released by the heart in response to stretching of the arterial walls. Its function is regulating the blood pressure and fluid balance. So the mechanism by which ANF induces natriuresis and diuresis. So what is the basic difference between them? Excretion of sodium in the urine is natriuresis and increased urine production is diuresis. They are involved with the membrane bound form of GC. And both nitroglycerin and preside are involved in the production of CGMP through soluble form of GC. So the increase in the CGMP levels causes relaxation, vasodilation, causing a therapeutic effect in cardiovascular conditions. Okay. So this was the image. If you see, we have 2 types, membrane spanning GC. Another one is the NO activated GC, that is nitric oxide. So who is the precursor of nitric oxide? Which amino acid is the precursor of nitric oxide? Do you know? Which amino acid is the precursor of nitric oxide? Any idea? Arginine residue. Remember this part. Okay. So you have got ANF, guanyline and endotoxin. They can serve as the ligand for the membrane spanning GC. Okay. Next, nitroglycerin. So as I told you, it's a vasodilator commonly used to treat angina pectoris, that is the chest pain as well as the heart failure. It gets converted to nitric oxide in the body, which is responsible for activating the soluble form of GC. So as I mentioned here, it is NO activated GC is given, right? Then once they are activated, CGMP will be released and it causes the relaxation and vasodilation. Same is the case with nitroprusside as well. It is commonly used to treat hypertensive emergencies. So they also get converted to nitric oxide, activating the soluble form of GC, CGMP production, thereby decreasing the blood pressure. Okay. Next question on your screen is regarding GT pathway. The sequence of events are involved in phototransduction, in rod cells, illumination, hyperpolarization, activation of phosphodiesterase, decreased release of synaptic transmitter, activation of transduction, decreased intracellular CGMP, and conformational change in rhodopsin. So identify the correct sequence of events in visual transduction during light perception. So regarding the GPCR, which I told you 4 different pathways are there. GS and GT is considered to be the most favorite for CSIR point of view. They often asked question from here. And that too, majority of times, they will be asking a 4 marks question. Identify the correct answer here. I mean, correct sequence over here. I hope so. You might have created some kind of flow charts or kind of images. You might have studied, right? Try to recollect and get into a conclusion. Okay, Anupam and Malini. So, is there anyone who would like to give the answer apart from Anupam and Malini? So, this is a kind of very easy and direct question. The only thing is that you have to remember the sequence through either a flow chart or through an image. So basically, signaling is all about remembering through flow charts or remembering through images. That's important actually. Others, try to give the answer. Very good, Abhinav, to get your response. Others, keep on trying. See, this is not an exam. Here you are testing your ability to get into a conclusion. So if you have studied it, I would like to say that do attend the answer. You are confused between 1 and 3. No problem. We will clear your confusion within short period of time, Malini. Okay. So the correct answer is 3 only. I hope so. The Malini, you got a conclusion over here. So what happens over here? Initially, conformational change happens in rhodopsin. So once rhodopsin undergoes conformational change, transducin gets activated. Once transducin gets activated, phosphodiesterase will be activated. So phosphodiesterase is responsible for breakdown of second messenger. So obviously decreased intracellular CGMP, right? And after that, hyperpolarization event takes place. Then there will be decreased release of synaptic transmitter. So you can remember in this way. First thing is that rhodopsin can be existing in 2 forms, 11-cis retinal and all trans retinal. The opsin protein is connected to this protein via lysine side chain. So when the light hits the retina, there will be an isomerization taking place, converting the 11-cis retinal to all trans retinal. And you will find that opsin is getting activated. This is how your rhodopsin is activated. Next, try to remember the difference between the thing, what happens in the dark condition, what happens in the light condition. So in the dark, rod cells are active. So CGMP is active, cation channels open, action potential, hypopolarization, that is nothing but your depolarization. That causes the release of inhibitory neurotransmitters. The nerves, which are nerve cells that are responsible for the vision and all, will be inactive because it is dark, and that's why you're not able to visualize in the dark condition. On the other hand, during the light time, rod cells are inactive. So with the help of phosphodiesterase, CGMP gets converted to GMP. Cation channels close. There won't be any action potential. That means depolarization will not be taken place, resulting in hyperpolarization. So excitatory neurotransmitters will be released. The nerve cells gets activated so that you are able to visualize the objects. Okay, so this is the primary thing to be remember. And next is the pathway. So you can divide the pathway into three segments. One is the from 1 to 5 you have got the, in fact I can say 6, you have got the light phase. 7th is the dark phase. 8 and 9 becomes the desensitized, or you can say the recovery phase. So as you can see, when light hits the retina, the 11th is retinal, all transretinal, it getting exchanged. Transducin GTP GDP exchange to get itself activated. Removes the inhibitory subunit from phosphodiesterase. So this leads to the degradation of CGMP. All the channels will be closed, preventing the inflow of ions, leading to hyperpolarization of the membrane. Because of continued efflux of ions, there will be reduction in the cytosolic calcium ions, because of which it leads to two things. On one hand, it will be inhibiting the phosphodiesterase, and on other hand, it will be activating the GC, because in the dark, what we have learned, we need to increase the ion channels. So once your CGMP is activated, ion channels will be activated, and there will be restoring or returning the membrane potential back to the pre-stimulus stage. Finally, if you want to terminate the pathway, what happens? You have got rhodopsin kinase phosphorylating the rhodopsin protein. Recoverin will help in stimulating the reaction. The phosphorylated rhodopsin will be attracted by another protein known as arrestin. After some time, arrestin will be released. Your all transretinal will be released, and rhodopsin is ready to take up another set of reaction by converting into 11 cis retina. So this is what the situation is when we go for GT pathway. Next question. Now this is a match the following kind of questions. They are provided with the ligands on column X, effectors on column Y. Identify the correct combination here. So nowadays a lot of match the following kind of questions are being asked in the exam. And I will always suggest that never skip the match the following kind of questions, because it is scoreable. You can easily fetch marks if you are able to if you are confident with certain combinations, because as you see, the terms are quite familiar if you have studied the life science syllabus. Serotonin, acetylcholine, immunoglobulin antigen complex, light. Terms are familiar. I hope so. Your confusion is gone. Malini is resolved. Now you will not think of option 1, I guess. Yes. Try to get into a conclusion to this question, answer to this question. Keep on practicing more MCQs on those topics which you feel you are weaker, because it varies from person to person. So depending upon your weakest portion, try solving questions more, try focusing on revision more. Yes, what could be the answer for this question? Okay, Anami, good to see your answer. Anami, Anupam, Malini. What about others? AAA and Abhinav Sharma. Would you like to take up the answer for this question? So you have to be a bit patient when you're trying to solve these kind of questions. And majority of questions that I have taken for you is from recent question paper. So you can see how the questions are coming in the recent past. And I always say signaling is a topic which if you study, you can easily carry Good marks back to home. Okay, Arzu. So let me get into a conclusion. Yes, and you all are correct. Option 3 is the correct answer. So basically, I'm pretty sure that you are confident with A, B, and D. And D, just now we had discussed about it, right? So D is 2. CGM, light is related to CGMP phosphodiesterase. So that you get into a conclusion. Serotonin is related to a ligand that works in the GS pathway. If you remember, you have a table in many of the standard reference book, the list of ligands with their corresponding signaling pathway. Do check on that. Acetylcholine. So acetylcholine comes in two ways. Either it can activate AC if it is your GS pathway, or it can activate the potassium channel if it is your GI pathway. Now, what is left is immunoglobulin E antigen complex. Now, this is a immunology related term. Well, it is related to phospholipase C. So, even though you're not able to get into a conclusion of one, but still, if you're confident of other three, can easily fetch a 4 marks out of this. See, allergen binding to the immunoglobulin E on the surface of mast cells or basophils. So there will be cross-linking of the receptors. So the ligand goes and binds to the receptor on the cell membrane. This leads to the activation of phospholipase C that triggers a signaling cascade inside the cell, activating the second messengers IP3 and DAG. IP3 will be releasing the calcium ions from intracellular stores, and DAG will be responsible for activating the protein kinase so that it can phosphorylate to generate the cellular response. Degranulation, the increase in the intracellular calcium and activation of PKC will be leading to degranulation of mast cells or basophils. So what it leads to? It leads to inflammatory release of inflammatory components contributing to allergic symptoms. So this is what the pathway of immunoglobulin E with respect to phospholipase C. So just a reminder to all of you is that our Josh batch is going to start from 1st of July, and you can join this batch. So this is your 2024 June 2025 batch. You will be having access to many things like AIMNet test series, 24/7 chat support system, then certain tools like mind maps, some AI generated AIMNet series are also available. So these all things will make your revision easier and quicker. That will save time just before the exam. So you can take the best use of this batch by accessing all these components. Then if you have any query, you let us know by calling in either of these two numbers so that we can clarify on that. Okay. Moving on to the next question is regarding bacterial chemotaxis. Okay. And here you need to identify the statement that is incorrect with respect to chemotaxis receptor. So receptors are dimeric transmembrane proteins that can bind to specific attractants and repellents on the outside of the membrane. Cytoplasmic tail of the receptor is stably associated with histidine kinase key A via an adapter protein, that is key W. Receptor and its associated proteins are all clustered at one end of the cell. Binding of an attractant increases the activity, whereas binding of a repellent decreases its activity. Now you need to identify the incorrect statement over here. So when you come across these kind of questions, you'll find that this part is something that you are already aware. Okay. And you need to read the question whether they are asking you the correct statement or the incorrect statement. So here they have specifically mentioned it is not true. So what could be the correct answer for this question? And when it comes to bacterial chemotaxis, although questions are less, but even if they will ask, they will be asking in the 4 marks. So far, they had asked in the form of part C only. Just be familiar with the sequence of events that takes place when the attractant is bound to the receptor and when repellent is bound to the receptor, and their movement. Of course, everything is given over here. See, when attractant is bound, smooth swimming counterclockwise. When repellent is bound, increased tumbling by rotating clockwise. So sometimes they can also ask you based upon their movement. So be clear with that. Yes, I can see Arzu, Anami, and Anupam has given the answers. Anyone else would like to take up the answer? Anyone else? The activity of receptor decrease. Why would the activity of, that's what the question is. We need to identify the incorrect statement here. Okay, so let's get into a conclusion now. And as you said, miscellaneous answer, yes, option 4 is the correct answer. So binding of the attractant actually decreases the activity of receptor, and binding of repellent will be increasing the activity of receptor. That's what the answer is. See, so you have cell extensions like flagella for locomotion, pilus for mating, fimbriae for attachment. Same is for glycocalyx, cilia present in higher organisms. So when a repellent goes and binds to the receptor is activated. The information passes to key W, that is an adapter protein, activating the histidine kinase, that is key A, with the help of ATP production, ATP hydrolysis, and that phosphate will be transferred to key Y. So they will help in the flagellar motor to rotate in clockwise rotation. So that results in tumbling. On the other hand, you can guess it out. If attractant binds, the receptor becomes inactivated. Phosphorylation events will be decreased. Whatever the phosphorylated intermediates are there, key Z, the phosphatase, removes that phosphate. So you'll get the dephosphorylated version that will help in movement of counterclockwise rotation. That is smooth swimming. Okay. This is how it looks. And why this happens? Remember, when the repellent binds to the receptor, key R will be adding the methyl group to the receptor, thereby activating it. So when key A is activated, it will add phosphate to key B on one side and key Y on the other. So that will result in tumbling. On the other hand, so when an attractant binds, we have the phosphorylated key B. This will remove the methyl group in the form of methanol. Okay. So the phosphorylation activity will be decreased. Then we have got the key Z for the conversion. I hope so you got your answer, Anupam. This is how the working principle of chemotaxis system. Okay. Next question over here. Prolactin is an anterior pituitary hormone which is lactogenic and helps milk production in mammals. Statements below are made regarding the mechanism of action of prolactin. Receptor that binds to prolactin lacks intrinsic kinase activity. Upon binding, it dimerizes and associated kinase gets activated. It leads to activation of its intrinsic kinase activity. STAT plays a role in the prolactin action. There is no involvement of STAT in prolactin action. Identify the correct statements over here. Now, this is a kind of question which can be expected that it might have come from unit 7. because it is about an hormone, endocrine hormone, or can be expected to say as unit 4 because they are asking you the mode of action of prolactin hormone. And I hope so. Anami and Anupam are clear with the previous question. Yes. Okay, Malini. Malini has given the answer. What about others? It's not that much tough, but you need to connect it with two different topics. And the major part is you should remember the ligands that participates in the signaling pathway. Other than Malini, anyone else would like to take up the answer? Anupam, Anami, Arzu, Abhinav, AAA. If you can rename, it will be easy for us to ask you. Triple A. Others? What about others? So let's get into the answer. The correct answer over here is ABD. Yeah, the thing is that you have to be clear with the ligand over here. This is a table that gives you an information that what are the signaling proteins that will be associated with JAK-STAT signaling pathway. And JAK-STAT signaling pathway is an example of receptor without intrinsic kinase activity, if you remember. Right? So that's why what happens in this case is that you have got ligand which goes and binds to the receptor. Receptor is not having the intrinsic kinase activity. So what happens? They will be taking the help of soluble cytosolic tyrosine kinase. Right. So this will be recruited to the receptor, undergoing autophosphorylation, transferring the phosphate to the target. And here the target is STAT. So based on that, the signaling happens. So the core information over here is you need to remember the ligand for different signaling pathways. Okay, I hope so. Asta and Arzu, you are clear with the question. Take up this question from cellular communication. So basically, that's the kind of questions you can expect from signaling part. When it comes to cellular communication, majority of times you will find that it will be asked in the form of match the following. In fact, recently also, last exam also, if you have seen, and that too, they will be asking from 4 marks question. Here a lot of proteins are there, junctions are there, connecting link is there. And you need to identify, well, these are examples of your cell adhesion molecules. So based upon their structure, based upon their function, they are going to ask you this question. Okay, Astha and Anupam. Anyone else would like to take up this question? So, well, integrin is a protein that can serve both as a cell surface receptor as well as your cell adhesion molecule. Has to be remembered. Cadherin, the unique feature is that they are responsible for conducting stable junctions. Then you have immunoglobulin superfamily, then you have got selectins. Different functions. Immunoglobulin superfamily, as the name indicates, is related to antibody structures, the corresponding receptors and all. So what could be the correct answer over here? The correct answer over here will be okay. Astha Anupam Arzu has given 4 as the answer. Any other answers apart from 4 that anyone would like to take on? Okay, I can see every one of you are giving answer as a 4. And yes, you are absolutely correct. So be clear with the functions. Always remember selectins is related to uses carbohydrate as their ligand. So lectins that can mediate transient cellular interaction specifically in the bloodstream. Catecherins, as I told you, they are important cell adhesion molecule as they form stable junction via strong homophilic interaction. Homophilic means same molecule will be interacting. Immunoglobulin superfamily, they will be having immunoglobulin like domains and required during development and regeneration. Integrins, as I told you, is an example of a cell surface receptor come CAM. So it is the only receptor into which your ECM proteins can go and bind. So ECM proteins will serve as the ligand over there. Got it? Okay, anyways, all of you has given the correct answer. This is a recent question. So here they have provided with the sustained statements like tight junctions are cell junctions connecting intermediate filaments in one cell to that of the other. Desmosomes are cell matrix junctions connecting actin filaments in one cell to that of the other. Gap junctions are channel forming junctions allowing the passage of water soluble molecules from cell to cell. Tight junctions are occludin junctions that seals the gap between the cell. Hemidesmosomes are cell matrix junctions connecting intermediate filaments in one cell to that of the extracellular matrix. Identify the incorrect statement over here. Again, the prime focus will be identification of incorrect statement. And if you are able to justify the answer, whatever you are giving, that clearly indicates that your concept is clear. You know which connections, which junctions are connected through which cytoskeletal proteins. Find out the incorrect statement over here. I got answer only from Astha so far. Anupam. Others, try on. This is again not a tough question. Very easy and direct question, 4 marks. So last time they had asked in the statement based question in one shift, and in another shift they had asked the match the following kind of question. So those of you who are giving the answers, try to mention why those are incorrect. If you feel A and B are incorrect, let me know why it is incorrect. Now that will help you to identify that your concept is clear or not. Okay, Arzu. Yes, you're absolutely correct. So basically, A and B is correct, and I mean, they are the incorrect statements for this question. So what should come over here? Why statement A is incorrect? B is incorrect. As you rightly said, they are cell to cell interactions. Hemidesmosomes are examples of cell to matrix interaction. Another important point over here. Are they connecting actin filaments or intermediate filaments? Are they connecting intermediate filaments or actin filaments when it comes to desmosomes? Remember, whether it is your desmosome or hemidesmosome, both of them connect via IF, that is your intermediate filaments. Why statement A is incorrect? Very good, Anupam. Yes, they are not connected by intermediate filaments, rather they are connected by actin filaments. That's why statement is incorrect. So now you are able to identify why the statement that your concept is clear or not. Simply identifying the answer is not the task. Identify why the answers are incorrect is a very important task. Okay, try solving the questions in this way. Another match the following kind of question for you. Now, this is related to your ECM proteins: nidogen, fibronectin, integrins, vimentin. Identify the correct combination here. So what kind of questions you can expect from cellular communication? You can expect match the following or statement based questions regarding cellular junctions, regarding cell adhesion molecules, regarding ECM proteins. It could be function, structure, anything they can ask. Wherever you find it bit confusing, first make yourself calm, try to recollect what you have studied and incorporate the answers. Try to get at least some combinations in your hand as correct. Some combination as correct. In that case, you'll get into an answer. Try it out, everyone. Is it tough? Do you find it tough to answer it? Astha and Anupam. Two different answers. How about others? Arzu. What happened, Malini? Is this part is not so clear with you? I saw initially you were giving all the answers. Yes. Sure, Astha. So this is what the intention of solving the questions. When you're solving the questions, you know that which part has to be revised. You need revision the most. Okay, triple A. Okay. So let's get into the answer if no one is else adding up the answer. The correct answer over here is 3, as mentioned by Arzu and triple A. So A to 3, nidogen is a principal component when I speak of basal lamina. If you remember, nidogen is also known as entactine. Okay. Fibronectin, yes, they can produce different proteins by means of alternative splicing. Integrins, it is a heterodimer. Why? Because it is a combination of alpha and beta subunits. And earlier itself, I said you, it serves as a receptor. So obviously, ligands will be your ECM proteins. Vimentin, this you might have studied under cytoskeletal system. One table is there. Types of intermediate filament. This comes under there. Type 3 intermediate filament. It is typically expressed in epithelial and mesenchymal cells. You need a revision of this. See, nidogen 1 and 2, both of them are ubiquitous components present in the basement membrane zone underneath developing the epithelia of most of the organ systems. Alternative splicing, you all might be aware from unit 3. So the fibronectin uses this mechanism to produce up to 20 proteins. You might be aware that you have few alpha and few beta subtypes. So they shuffle among each other. As I told you, vimentin is an example of a type 3 intermediate filament. Okay. So, another very rarely, but sometimes you can expect experimental questions from this part. Cadherin mediate calcium dependent cellular interaction and plays an important role in embryonic development by changing the adhesive properties of the cell. Aggregation of nerve cells to form an epithelium is correlated with the appearance of N-cadherins on cell surface and vice versa. NCAM, that is your neural cell adhesion molecules, belongs to immunoglobulin superfamily involved in fine tuning of adhesive interactions. So, in order to study the effect of N-cadherin and NCAM, two sets of mice were generated. Set A is the mice with mutation in N-cadherin and set B is the mice with mutation in NCAM. You need to identify the result that is likely to happen because of this two mutation. Very rarely, in fact, if you take up the total number of questions, these kind of experimental questions are very less asked, but if they are asking, again, it is in the form of 4 marks. The fact regarding unit 4 is that you'll get a lot of questions and majority of them will be in the form of part C. It holds a good weightage when I say examination point of view. So coming to this question, you just require the logic. Even if you don't know what is a cadherin, I mean, you are not able to recollect what is a cadherin protein is. Simply by reading out the question, you can get into a conclusion. Now, let me see who can do that. Malini, got your answer? Anyone else? The answer is present in the question itself. Always remember one thing, whenever you do experimental questions, I'm not saying always, sometimes you can get into a conclusion simply by reading the question properly and thoroughly. And this is just one example of that. Apart from Malini, anyone else would like to take up the answer? It's not difficult. It's easy. The reason is that you have, repeatedly I'm saying, you have the answer in the question itself. Just read it thoroughly to get into a conclusion. And whenever you face these kind of questions, you can star mark it to review it later, so that once you have done with the questions which you know, you can have a look onto this patiently if you think that it's prolonging for you to get into a conclusion. Okay, Malini, Anami and Asta, let me give you the answer. Answer is 2. Very clearly it is mentioned in the first sentence itself. Cadherins will be mediating calcium dependent cellular interaction, plays an important role in embryonic development. So N-cadherin involved in that. So obviously if this protein is mutated, the mice will die in the early development because cadherin is required during the early development, embryonic stage itself. But on the other hand, if you see the NCAP, what they are responsible for? Fine tuning of adhesive interactions. So they are required for similar interactions. For example, if it is a nerve cell, then nerve to nerve interaction. If it is a heart muscles, heart to heart interaction. So they are not affecting on the embryonic stage, but yes, they can develop abnormalities during the development of that particular system. Got it? This is how you need to get into a conclusion. Moving on to another topic, that is apoptosis. This is a recently asked question, I think last year. Apoptosis refers to PCD that is triggered by specialized intracellular proteases. We call it as a caspases. So it is clearly mentioned intrinsic pathway of apoptosis. This depends upon the activation of Fas domain by Fas ligand that activates the disc, regulated by BCL2 family of proteins. Key regulatory protein is APAF1, part of ETC. Recruits procaspase 9 to apoptosome, and once activated, they undergo cleavage to activate the executioner caspase. Yeah, it would not be survive means, see, you can differentiate a person, a normal person, with a mentally retarded person. So that is what the meaning is, Astha. A mentally retarded person, he is surviving, but is having the problem of mental retardness. So you can correlate in that way. I hope so. It's clear to you, Astha. This is again a very good question to get into a conclusion. Okay. So since we are approaching the time, so I'm getting into an answer directly. If anyone would like to answer, keep on answering it. It's for your revision. Correct answer is B and D. In fact, this is intrinsic pathway of proteins, right? So you can easily exclude the fast ligand part. Then it is a part of BCL2 family of proteins. Okay. And if it is 9, obviously it is intrinsic kinase pathway. If it is mentioned as 8, it will be extrinsic pathway. So this is A, and if I say 8, it will be a part of extrinsic pathway. Okay. So this is what the answer over here. Next question. Identify the correct sequence of events that occurs in the cell death pathway. Identify the sequence of events that occurs in the cell death pathway. BCD option 2. I need to check on that. So how you can approach this question? You can approach, yes. All you need to remember is, remember this flow chart. Who is the regulator? Who is the adapter? Who is the effector? So regulator is the controlling factor, synonymous proteins. Set 4 is the adapter, like your APAF1. And set 3 is a effector, like your initiator caspase and executioner caspase. So based on that, yes, you'll get the answer 4. Okay. So EGL1 is with response to C. elegans. The gradient of oxygen is available to cells at inner regions of a tumor tissue environment, typically low, that creates a hypoxic microenvironment. If enough oxygen is supplied to the cancer residing in hypoxic microenvironment, which of the following processes will not occur? HIF1 alpha stable in the cells under hypoxic condition will undergo oxygen dependent hydroxylation targeting for ubiquitination. Warburg effect will be reversed and conversion of glucose to lactate will not take place. Expression of HIF1 alpha dependent genes will be lowered. Lactate generation in tumor microenvironment, which contributed to M2 polarization of tissue associated macrophages, will continue. Identify the incorrect answer from here. This is also a recently asked question. So how you can approach this question? Actually, this question is very simple if you just know one term. See, the correct answer over here is Warburg effect. Because when it comes to cancer cell, Warburg effect is increasing. Okay, so this, even if you're not aware of statement 1, 3 and 4, if you know what is a Warburg effect, you can easily fetch a 4 marks out of this. Here it is not reversed. Here it is accelerated over here. See, Pasteur effect means glycolysis can be inhibited by high level of oxygen, but Warburg effect means even if the rate of glycolysis is happening, it is independent of presence and absence of oxygen. So that is what we call it as a Warburg effect. See, if you compare these two, differentiated tissue and a proliferative tissue, their preference is oxidative phosphorylation. But when I say the proliferative tissue, they will start regenerating lactate because they will provide the carbon skeleton required for the glycolysis. From the glycolysis, they will be generating. So this will regenerate the NAD plus for that. So the carbon skeleton can be used for the production of biomolecules over here. That is the fact. Okay. And the last question over here is, following statements are made about some of the abnormally expressing proteins in human cancer. Increased telomerase expression always contributes to increased cell death in cancer cell. Overproduction of BCL2 leads to inappropriate cell survival, leading to CLL. EF, E6, E7 encoded by HPV are tumor suppressors. Overexpression of cyclin D1 or loss of P16 and RB can cause inappropriate and unregulated passage through restriction point in late G1. So what is the combination of statements that are correct? So the correct statement over here is B and D. Okay. And why A and C are incorrect? C. Increased telomerase expression means increased number of cell division. Now that is the feature of cancer cell. Similarly, E5, E6 and E7 are not tumor suppressors. These are oncoproteins. Okay. So that is what the answer is. And yes, we have done with the session. Just wanted to mention one thing. Know well what leads you forward and what holds you back, and choose the path that leads you to the wisdom. Follow this in every aspect of your life. You will definitely achieve what you are dreaming of. With this, yes, thank you so much for attending the session. Keep on practicing the question. I will see you again in another session of Brahmastra series in the upcoming days. Thank you so much.